This PowerPoint is on protein making organelles in the cell. There is another PowerPoint that's on non-protein making organelles. Organelles that make proteins are a system. They work together, and they're kind of like an assembly line. They make the product, they fix the product so it's right, they wrap the product and ship it off to somewhere else. So this process of making a protein, it starts with the storage of a recipe, and that's stored as DNA in a nucleus. This cell up here on the right is a melanocyte. This is a cell that makes melanin, which is a protein, and it's also a pigment in your skin. So the process starts when we access the recipe for the protein. And we use some of these organelles to interpret the recipe of the protein. And in doing that, we make an assembly of amino acids, which we know is the first level of protein structure. If you need to, you should go back and review that mini lecture from a previous module. We're also going to fold the protein, right, because it has a specific shape, and then we're going to ship it somewhere. We might keep the protein in the cell itself, or we might release the protein, like this melanocyte here is releasing melanin granules to the cells around it. It depends. Every protein has a recipe for making it, but that recipe also contains information about where the protein needs to go. Okay, so I said everything starts in the nucleus, which doesn't necessarily have to be at the center of the cell. It can be off to the side. But this picture here, let's start looking at it at the top left corner of the picture. It's showing us a cell, and it's saying that the nucleus is that colored sphere in the center. So we say that the nucleus contains your genetic material, and just as a little hint there, that's DNA. It does have its own envelope. It is not a cell, but it has a lipid membrane around it. And this picture on the bottom of that insert there, that's a pore. And that pore is only big enough to allow certain substances in and out of the nucleus. So DNA is actually too big to get through the pores, and therefore DNA never leaves the nucleus. If your DNA does leave your nuclei, you have a problem. You should see a doctor. There are no nuclei in red blood cells. In fact, red blood cells, as they develop, they puke out, throw up, or exocytose their nucleus. And they do this in an effort to make more room so that they can carry oxygen. This also means that red blood cells cannot regenerate themselves, which is why they're recycled via your liver and spleen about every 4 months. There are lots of nuclei in white blood cells. Sometimes they can have multiple nuclei. Sometimes they'll have a nucleus that has all of these lobes in it, like a flower, and the nucleus will get larger as you age because your immune system encounters different pathogens or invaders, and it stores the information about those pathogens as DNA in its nucleus. So as you meet chickenpox and COVID, hopefully we can develop immunity, but as you meet them, a white blood cell will store information about how to attack them if they should ever encounter them again. I have other PowerPoints or mini lectures about mitosis and protein making that help you understand some of the different words about genetic material. But I just want to make the point that we have different words to refer to humans: child, adolescent, adult, geriatric, right? And these refer to like an age, but they also refer to a shape and a size of humans that's an average for us. So genetic material, or DNA, is similar. It has different names depending on the structure that it's in. The most familiar name that I want you to get to know is a chromosome. That's just a piece of DNA. That's all it is. You have 46 of them. They come in pairs. Anyway, DNA can also be called chromatin. This means that it's making proteins, it's in the nucleus, it's doing its job, it's being active. So this is a little bit more of a specific word that tells you that it's DNA, but also what it's doing at the time. And chromatin looks like spaghetti over here on the left. A chromosome is very condensed, and you can see a chromosome over here on the right. These two things, they're both DNA. Both of them are DNA. And that's what this picture in the center is showing you. It enlarges, 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 and it shows you DNA. Okay, so right outside the nucleus, because the nucleus has the recipes for making a protein. Right outside the nucleus is the machinery for making the protein, right? So it's like the kitchen, right? The recipe stored in the cookbook, the nucleus, and this is the kitchen. This thing, a ribosome, it's going to assemble amino acids from something called RNA. Some of you are like, oh, I remember that. Others of you may have to view the transcription translation mini lecture on making a protein. Ribosomes can be free-floating in the cytosol. They can just be anywhere, right? But most of them are actually fixed to another organelle that I'm going to talk about in the next slide that we call the ER. And you can see that picture up here on the right. It's showing you a free ribosome and then those that are attached to an ER. This determines kind of the location of a protein that they make. So there are lots of ribosomes in cells that make proteins, right? And we talked about a melanocyte that makes melanin, which is a pigment protein. So they have lots of ribosomes. Your liver makes liver proteins that are important for clotting in your blood. And it has liver cells have lots of ribosomes. So if your job is to make lots of proteins, you have lots of ribosomes. So I mentioned this, the endoplasmic reticulum. That word is terrible. We never use it. We call it ER. But what the word actually means, network inside the cell. That's all it means. And its name refers to the fact that it looks like a maze or a network, and it is inside the cell. The word reticula means network or webbing. So the endoplasmic reticulum is usually right outside the nucleus with the ribosomes. You can see that in the picture up here on the right. Let's look at the picture down at the left, down at the bottom, starting on the left. We see a picture of a nucleus, and we're being shown an enlarged section of the endoplasmic reticulum. And it does look web-like or maze-like. And you can see ribosomes attached to some of it. So there's the rough ER and the smooth ER. And this is what we're concerned about. In general, they both participate in folding a protein. So the protein's being made by the ribosome, and then it's being folded by the rough and the smooth ER. So the rough ER is called as such because there are ribosomes attached. There are lots of these, lots of ER and enzyme making cells. So enzymes are proteins, and your pancreas is the organ that makes an excessive amount of digestive enzymes. And therefore, there's lots of rough ER in pancreatic cells. The smooth ER is called such because it gets all the girls. No, I'm kidding. I'm sorry. It's a bad joke. The smooth ER is called as such because it doesn't have ribosomes attached. It does look like more of a webbing, and the rough ER, to me, looks more like a maze. The smooth ER likes to make lots of glycolipids. These are big molecules. We mentioned them. They like to cover the outside of the cell. Glycolipids can also be used to tie up toxins and then remove them from your body. And many of us know that your liver does detox. Yes. And the smooth ER makes glycolipids that can bind up the toxin. Therefore, your liver has tons of smooth ER. The Golgi apparatus complex body. I have heard so many names for this thing. You can call it whatever you want because I can never remember what class I'm in and what the textbook calls it. As long as you're using the word Golgi, I'm good with that. So I'm sorry. What the Golgi does is it receives the protein from the ER and then it determines like where it's going to go. It's like shipping and packaging. Look at this picture down on the bottom left. The ER is up in the top left of that picture, ribosome is attached. And look at how vesicles are coming off of the ER and being received by the Golgi, which is the green. And then the Golgi kind of packages it up, puts a bow on it, slaps the shipping label on it. And then the Golgi can release the proteins in vesicles that are puked out by the cell. You can also see the sequence up here in this melanocyte. Nucleus, ER, Golgi, vesicle. So, like I said, it's an assembly line. When the Golgi makes vesicles, one of those fates is that it can be puked out by the cell. But vesicles from the Golgi have three fates because the Golgi has other responsibilities than just protein making. So yes, number one, the Golgi can make a vesicle that's full of the protein and the cell will puke that out, and that's called exocytosis. Number two, the Golgi can make a vesicle that fuses with the cell membrane and repairs certain areas of the cell membrane. And you can see that down here. It's labeled as number two in the picture. The third fate is that the Golgi actually makes those lysosomes. Lyso meaning to split or destroy. There is another mini lecture that tells you that lysosomes are like little tiny stomachs that are mobile in the cell. They work and they do digestive processes. So the Golgi makes them. All right, so let's take a step back and look at a picture of a cell. I like this picture. It's nice and simple. The one in your book is overly complex to me. But it's an assembly line, right? We assemble the package of protein, fold the protein, ship the protein, and there's many ways that this assembly line could malfunction, right? One of the workers could refuse to come to work one day. They could do their job poorly. The Golgi might ship the protein to the wrong place. All of these things that happen, they result in diseases. This is the guts of a disease, is how proteins kind of function in your body. So we've been talking about cystic fibrosis, and there are actually many ways that cystic fibrosis can work. And cystic fibrosis can occur from a lot of different malfunctions in this assembly line. The protein might not be folded correctly. The protein might not make it to the cell membrane in the right place. So you have to think of what each organelle does and how it could malfunction, leading to the non-working of a protein. Oh, there we go. So an example, the Golgi doesn't make vesicles. The protein is never released from the cell into the body. I like this picture. It labels all of the organelles for you, some that are protein making, some not. And it has a nice little statement at the top that says the endomembrane system is a protein highway, right? And it's assembly a line along which proteins move until they're embedded into their final destination.